So I'm also a statistical physicist and a biophysicist. One of the benefits of seeing a few of us is you get to see the variation within the type. And so it's a little bit of a different talk. You can see the background. I have a network, so I'm interested in networks. I could have been in the network session. I'm going to tell you a bit about stochasticity. I'm going to tell you about dynamics, a little bit about methylation clocks. But this is work done largely by Glenn Pridham, a former PhD student, now with Yong at Weizmann, and a little bit by Spencer Farrell, another former PhD student, and in collaboration with Kenneth Rockwood, a gerontologist, and Karthik Tenenkor, a nephrologist. So we're a gerophysics meeting, but for the physics perspective, you could say it's a subfield of physics. And so it's the physics of aging. And so reflecting on what that means as a physicist, for me, I'm very engaged with dynamics as a physics background. Maybe you remember in first year physics or in high school. So if you want to get at the dynamics of aging, and clearly aging is a dynamical process, one of the questions you want to do if you want to look at it quantitatively is to understand the variables of aging, what the variables you want to do. And if you have more than one of those variables, you want to look at the interactions of aging. We saw an example in the last talk about the utility of looking at interactions. And so really, this is also in a complex systems perspective that rather than picking, for instance, one clock or another clock, you want to look at all the clocks that you have and look at how they interact with each other and how they evolve dynamically. So if you want to do that, and largely this isn't about methylation, so when we started this, we were looking at standard blood tests because those are available in longitudinal studies. If you want to look at the dynamics and you want to build a model, you have some desired model ingredients. So this is I guess you could say a principled approach, but it's more of a phenomenological approach, but you still have to look somewhere. And one thing you want is a dynamical model. You want something with time dependence or age dependence. The other thing is you want to include stochasticity because clearly there's a lot of variability even within an individual. You want to include interactions. And finally, you'd like some ability to capture homeostasis. So my blood pressure today and my blood pressure a year from now and my blood pressure 5 years from now isn't varying all that strongly. So there may be weak instabilities, but certainly you want to not build in the instabilities or not build in, you want to allow for stability in the system. So when we started this, we started with a kitchen sink approach. So the kitchen sink approach was sort of a deep learning approach. Well, let me just look at this equation. This is the change of a particular value, say value I of n different biomarkers, and it's represented by some arbitrary function which is captured in a deep neural network, and noise which is captured in a deep neural network, but a very explicit interaction term where a different variable pushes changes in this variable through this interaction matrix W. So we put in a constant linear, in the physics approach of keeping things simple, constant linear interaction. Now, and this was work done by Spencer Farrell. Now the interesting thing about this and why I want to mention it is we also, Spencer also did the same dynamics, but instead of that explicit interaction, just embedded it in a general deep learning approach. So the deep learning approach just said, whatever the interactions are, nonlinear, doesn't matter, we'll just build it all in. And horse raced the two models. And so there's some details of the arbitrary interaction. It was in a latent space we could tune the dimension. But the main thing is that the top model with the explicit, very simple interactions is the red dot. And the bottom model, which has these arbitrary interactions, arbitrary latent space, is the black dots. And basically they're the same. And what that told us was that these interactions that we had in the biomarkers that we were tracking were very simple. They were age independent, they were linear, and they were not only age independent, but of course if it's age independent, they were health independent. So in a way, this is not surprising. You know, when I give this talk and people are close, I say, you know, punch me in the arm. Maybe I'll do it. Yeah, so Max doesn't hate me, right? And, but he punched me in the arm. And that's basically, we all know that we're very stable systems, right? So he can do that. I'm not a house of cards. I'm not going to fall over. The interactions we have between these variables, at least at higher scales, are pretty weak. We don't expect blood sugar to strongly affect blood pressure in a dysregulated way. So with Glenn Pridham then, we took that insight and built it into an explicit, without the deep learning, very interpretable dynamical model. And on the left, so we have a vector. There's a physics notation. This is a vector, I guess, in a deep learning as well, of all the measurements. So there might be 20 or 100 of those measurements. The index n is the time points that are measured. So the left-hand side of this equation is the change between observations. So this is the dynamics. We have a noise term, this epsilon. We have a fixed point and an interaction matrix. So changes are driven by different deviations with respect to the fixed point. Interactions are driven through an interaction matrix. So from a physics perspective, you could say, well, this is a Taylor expansion. We're taking a low order interactions. We have a constant term and a linear term. But, and that's true. And you could also, with the addition of an explicit time step, see this as just a very simple differential equation covering the time dependence. The nice thing about it is it's so simple that it's linear. So if you look at all the y's in here, they only come in once. So you can use very simple linear statistical techniques to obtain the parameters in the model. So linear regression with covariates. And it's important to recognize, okay, where anything not in the model is going to be captured in the noise when you're comparing it to real data. Implicitly, you're looking at slow variables. So you're not, for instance, doing a circadian rhythm or something like that, oscillating. You're looking at stable things. And so this is an embodiment of the concept of complex aging, that any one thing you measure, one of these elements of the vectors, could affect anything else. And recapitulating the deep learning, we found that this interaction matrix and also the noise distribution were age and health independent within the quality of the data. This is 3 different publications and one on archive. And I'll give a link at the end. Okay, so what does it look like? Well, the raw model has a largely diagonal. Blue is negative, indicating stability, but some off diagonal with positive feedback with the red. And you can find a diagonalizing transformation. This is a linear transformation, so this is something which is invertible. So you can go from the raw observed values to this diagonal representation and back. So it's a lossless, you've not lost any information. And in that diagonal representation, that interaction matrix is diagonal, of course. It's all stable. So in all of our studies, we find stable. And in this space where you have these simple interactions, where you don't have any interactions between variables, we call it natural variables. And from a physics perspective, these are eigenmodes. It's a standard linear algebra. So nevertheless, so here's the equation with an eigenmodes, simplified because you have one rate for each mode, and you have a stable point, and you still have noise, so it looks the same, but they're not coupled anymore. So you can look at it, for instance, rank order the stability of these natural variables. So for instance, the initial study was with ELSA data. We also looked at some mouse studies and some dementia. The longest time scale was a lifespan. So our slow time scales getting at the idea of, what's the slow time scale. You certainly have slow time scales on the order of the life scale of the mice or the humans, which we capture, but there's a big range of time scales that you capture in the data. Nothing unstable in our analysis. So if you then, and this is from the kidney data, but if you then take these natural variables and do a proportional hazards model to capture how the particular variable contributes to the hazard of mortality through this beta coefficient, then plot those beta coefficients against something. So for instance, this time scale, the stability, we find that the slowest variables, these are the ones with the weakest stability, the largest lambda inverse, are the highest hazard coefficient. So these slow variables are unhealthy, sort of reasonably enough. But it's sort of a double whammy, because not only are the slow variables unhealthy, but when they have a large value for an individual, if I had a slow variable which was a high value, it would be slow to recover. It's not stable, so it's unhealthy, and it's unhealthy for a long time. So we call that stochastic accumulation. Now you notice that there are some points which are way off this regression line. So if you plot them in the hazard versus something else, you might capture it. And we found that if you plot it in the hazard beta versus the drift rate, this is the, remember I say we could put in covariates in the linear regression, so we put age in as a covariate. And we found that some of these variables had these fixed points which drifted with age. So for instance, my blood pressure ramps up slowly as I get older. Most people do, but my blood pressure is stable any given day. So that age-dependent drift is captured in this mu t. We plot it there, and those outlier points, the blue points, now fit better on this noisy regression line. So here we have that variables with age-dependent drift are unhealthy. You notice that they drift in positive direction, the beta is positive, negative direction, the beta is negative. They're all drifting in the unhealthy direction. So not only are strongly drifting variables unhealthy, but they're strongly drifting in the bad direction, and so we call that malastasis. So at least in this phenomenological approach, this is how we see these dangerous age-dependent variables coming out naturally in a fairly simple statistical analysis. So this is a session on stochasticity. So when you diagonalize the variables, the interactions, you're not diagonalizing the covariance matrix. You're left with a close to diagonal covariance matrix. These are the correlations in the residual noise terms in these variables, but they're block diagonal. So it's not a mess, it's pretty diagonal, and there's these clear blocks. And you notice that the blocks are small along the diagonal, and that means that the interactions here are basically between variables that are at the same time scale, the same stability. And so we see these are natural noise modules that appear to be dynamically coherent. Now, within the model, I just put it down at the bottom, the noise is all the misfit from the analysis. So anything that we haven't put in, and all we've put in is basically, the homeostatic regulation of the variables. Anything else isn't there. So for instance, if I get run over by a car, it's not in the model, it's going to be captured by the noise. If I go to the doctor, it's not in the model, it's going to be captured by the noise. So all those extrinsic effects are captured by the noise, and this is saying that those extrinsic effects seem to be coherent and ringing the bell, as it were, of these modules which seem to be dynamically coherent. So we see that the noise looks interpretable. It doesn't look like junk. And at least in the kidney study, we could then correlate these strong, slow modules with mortality outcomes and syndromes for kidney failure group people. Okay, so here's a summary. I'm not sure where I am for time. So the question was, I have a couple more slides. So what are the dynamics of aging? Well, we say, well, okay, at least for these sort of variables, we want to capture, stochastic homeostasis, and this is a natural way of analyzing it that's light statistically. But we don't want to just do that in the raw. We want to make use of linear algebra and diagonalize this and capture these natural variables, which are linear combinations of the raw ones. And in terms of these natural variables, slow variables, weakly stable variables are bad. Drifting variables, age dependence, fixed points are bad. And the stochastic effects, the extrinsic effects seem to be coherent. In terms of the interactions of aging, well, explicitly we have these slow linear age-independent interactions, W, and implicitly, you're interacting with the outside world, and those interactions, at least it's not a physics interaction, but it's a language interaction. Those interactions are captured, for instance, through extrinsic effects in the Z variables, as I saw in the noise. Okay, so there's all sorts of questions that arise from this, and hopefully you'll ask me some of them when I'm done. But I'll start with addressing 3 questions that came to mind for you today. So the first question is, well, we're measuring, say, 30 variables when you take a blood test. What about the 100,000 odd variables that weren't included in the analysis? And so you see within this simple model that if you take a particular thing, say your serum albumin, and see how it changes, say, blood pressure, all those things are pushing one variable to another through this W interaction matrix. But if you don't observe it, it can't be included in the equation, so it has to be included implicitly somewhere, and one place it can be included implicitly is through this fixed point. So these fixed points, and for instance, the age-dependent drift of these fixed points is one place where unmeasured or hidden variables can include themselves in the model. I've also alluded to the fact that, of course, things that aren't in the model could be just captured by the noise. And that's another possibility. Okay. Second is how connected is this to PCA, or principal components analysis? And the answer is it's sort of connected. It's close in style, let's say, and actually we used PCA as a pre-processing step. But the important thing is that PCA diagonalizes the covariance matrix and figures out a transformation of the variables, so you get these different PC components. We're not doing that. We're taking the dynamics and diagonalizing the apparent interactions between variables. So we have dynamically independent, but then within those dynamically independent variables, some covariances, and I alluded to the fact that the thing that correlates the variables are these extrinsic effects in this picture. So it's because the covariance is simple, you can sort of have your cake and eat it too. You can imagine that the things that are driving correlated fluctuations that are captured in PCA are just these underlying biology in the system that we're largely capturing in this analysis. Okay. Last, to get to aging clocks, what about other slow variables? So I didn't really talk a lot about what variables we were using. I say blood variables, but Glenn also analyzed some data from the SATSA study, the longitudinal Swedish twin study, and looked at different aging clocks. And you can say, well, why are aging clocks stable? Well, if they're calibrated against something roughly, which is age, then you'd expect that they aren't going to diverge strongly from age. And so you can see here we have a color scale from molecular to organismal, and you can see that these things, these individual ages are stable, but you can also see that they're pushing each other. And so this can get at saying, well, let's take all these ages and see which age is pushing another age in terms of, in a sense, causal pushing. Now, underlying these are some biological processes you don't know, and it's a question of epigenetic clocks in general. You don't know if you modify them, whether you're modifying the underlying biology. But this can give you a way of looking at that system. Okay, so that's it. Time is up. So that's a QR code for the publications that I talked about and other work that we're doing in aging biology as well.
Thank you very much. Yeah, we have a couple of minutes for questions. Any questions?
Yes. Thank you, Andrew. Great talk. I'm interested, what data set you used for obtaining those results?
Can you speak a little louder?
What data set you used for obtaining those results?
Yes. So there was the ELSA data. So that's English Longitudinal Study of Aging. So that's a national longitudinal study. We had some, the twins data was the SATSA data. It was out of a paper from Hogg's lab. And we have some in-house data for kidney failure patients with, I didn't tell you about the details of that, so it was very high quality data because people were on dialysis and getting blood work done every 6 weeks. Yeah, but you can...
Is this data set open?
The ELSA is for application, the kidney data is not. Mm-hmm. Yeah.
Thank you.
Yeah.
Okay, thank you for a great talk. I really tried to understand your talk, and then also at the end, you put a lot of variables inside the model, and then how your aging dynamics looks like at the end.
Okay, this, for instance?
Mm-hmm.
So what does it look like? So this is the aging dynamics. Is that?
Maybe can you explain more biological way? Sorry
biologically.
about it. Yeah.
Right. Yeah. So biologically, it's more phenomenologically. So we're not getting at some, atomic or molecular scale. So we're simply saying these larger scale measurements, like you'd get out of blood work, seem to be homeostatic, seem to be returning to some stable values. And so this is simply representing how a change in, say, blood pressure could be related to how far away blood pressure is to some stable value and allow for these variables to affect each other. So biologically, we don't say necessarily that blood pressure is causing changes to, say, serum albumin, but there might be something underlying the biology that could be affecting them and mediating that interaction.
Okay, you are saying, so for example, you are saying the blood pressure is, for example, to understand aging dynamics with this equation, right?
Yes, we can capture for anything that you measure that's slow enough to be captured by these dynamics, so not day-by-day variation or hour-by-hour variation, but things that change slowly that are significant for, I guess, gerontologists or doctors. How do you capture that homeostatic regulation of your physiology? And how do you allow for the fact that these measures interact with each other in a complex way? And so this is the simplest way we could come up with that they could not only change through these fixed point values, but could also change each other through this interaction matrix. So we didn't build the biology in. We didn't say this variable affects that variable. We don't have any of those mechanisms. We simply said, what does the data tell us about what is a leading indicator and what is a lagging indicator in a sense? So, and that's all captured in this interaction matrix.
Okay.
Yeah, so with better quality data, you can sort of do a better job at getting the parameters, these interaction parameters, W and the drift and the noise. But what we have so far from the 3 studies that we've done is a fairly coherent, everything seems to be behaving coherently and as I described.
Okay.
Thank you very much.
Thank
Yep.
you.